Alright, this video will go with getting at chapter 20 here, 20.1 to 20.3 on phylogeny. So, phylogeny has to do with organizing life in some sort of systematic way based on evolutionary relatedness. And usually we draw like a family tree or a phylogenetic tree that'll look a little something like this. Or, you know, it'll be kind of like I said, a family tree with the organisms all represented as lines on this tree. So that's what phylogeny is, and we're going to talk a little bit about phylogeny today. So, two things really related are taxonomy and phylogeny. Phylogeny is the evolutionary history of a group of organisms, in particular depicting. So, depicting that relationship visually. It's represented in phylogenetic trees, or we'll talk about later cladograms. So, this is an example of a phylogenetic tree, and how it's kind of split up. So it's showing different organisms, and at every node or bifurcation is a divergent evolutionary event. So here, a common ancestor, these two organisms, the badger and the otter, common ancestor diverged into the two species that would become the badger and the otter. Taxonomy is the classification of organisms by morphology, so by what structures or physical features they have in common. And this is what Linnaeus did. Linnaeus tried to order all living things into different groups that he called taxa based on their physical similarities. And taxonomy is hierarchical, so it follows a hierarchical pattern. We have different groups. So at the highest taxonomic group, or taxa, is the kingdom. So for example, we have kingdoms of organisms. So we either have eukaryotes, eubacteria, or archaeobacteria. So those are the three domains. Then we have kingdoms: Plantae, Animalia, Protista. Those are kingdoms. Then we have phylums, classes, orders, families, genii, and species. So each group, as we go down this chain becomes, or this pyramid becomes more exclusive, fewer organisms, and the organisms in each successive step, all from kingdom to species, have more and more specific or detailed aspects of their morphology in common. So, for example, in the kingdom Animalia, all these are animals. However, in the class Mammalia, all of these organisms are mammals. So there's more specific similarity. Each level becomes more specific in what criteria its members have to be part of it. So each of these taxon are groups. So Chordata would be a taxon within phylum, or Carnivora would be a taxon within order. So taxonomy, looking at this previous picture, is one way we can make phylogenetic groups, but it's not the best way. It's not the best way. We'll talk a little bit more about that, but first we're going to figure out how to analyze or look at a phylogenetic tree. So branch point is any of these bifurcations. It shows us where lineages or common ancestors diverge, a speciation event, into different species. Next, there's sister taxa. So two sister taxa in a phylogenetic tree are just two taxa that are right next to each other that share a immediate or a very recent common ancestors, like B and C. A and G would not be sister taxa. They share a common ancestor way back here. A basal taxon is a species or a group that diverged very early in evolutionary history. So T or the G would be a basal taxon. It diverged very early in the ancestral lineage and then remained basically how it is today the whole time. No other divergent events. This x-axis here, we think we go from left to right. This x-axis here is time. So as time is going on, this is the present. And then back here, this is the past. One thing to remember, you cannot infer absolute time or relatedness with these trees. So for example, these branches aren't necessarily proportional. Putting this dot here as arbitrary, it doesn't mean that taxon B and C arose in the midpoint between the common ancestor and present day. Doesn't say that. We can't infer absolute time. We can't say that by the length of this line we know this happened 5 million years ago and this happened 10 million years ago. And we can't be like, all right, this seems to be halfway in between present day and the ancestral lineage, so we can't say this bifurcation happened halfway in between present day and the ancestral lineage. So those things we can't say. This is just merely representational. It's not very quantitative. How do we construct phylogenetic trees? So we can make them by looking at homology and analogy, both structural, having to do with physical, morphological characteristics, so shapes, and molecular. So like protein amino acid similarities, and DNA similarities. So both morphological and molecular homology is used to establish a phylogenetic tree or relationship. Differentiating homologous and analogous structures is really important when looking at morphological and homological trees. So for example, just because these two moles or types of mole look the same, so they are homologous morphologically, doesn't mean that they are very closely related. In fact, these two organisms are pretty distant, diverged millions, like 20 million years ago, pretty distant relatives. So how can that be? Well, convergent evolution, like we talked about last time, they have analogous structures. They look similarly because they've developed in similar environments. Like a shark looks similar to a dolphin, which looks similar to kind of like a penguin, the overall shape, because they needed to be streamlined for living in a similar environment, the water. But if you look at the actual bones in their appendages, they have an extremely different structure that represents a different evolutionary history. They look similar not because they're closely related, but because natural selection pushed them to evolve into a form that was similar based on a similar environment. However, Organisms that have homologous structures, or they might be used for different things, like an arm and a cat leg and a whale flipper and a bat wing, they all are structured similarly. That shows close evolutionary relationship. Even though they're used for different things, this overall body plan, bone structure is indicative of evolutionary relatedness. You can think of bats and birds. Bats are more related to other mammals than to birds, even though they both can fly. Flight developed or evolved independently in both birds and bats. Bats use these stressed membranous wings. Birds use feathers. And we can see that by their overall structure. Those are analogous structures, not homologous structures. We can also use molecular evidence, like you did last time with amino acid sequences. More similarities in amino acid sequences means greater or closer evolutionary relationship. Like we saw, the green monkey and the rhesus monkey, only one in the p53 protein sequence, amino acid sequence, only has one difference compared to a human. A chicken has many more. That's why the green monkey and rhesus monkey share 95% similar amino acids in the p54 protein. But the Congo pufferfish or the chicken share far fewer similarities. All right. One extremely important form of phylogeny is called cladistics. And why cladistics is important is because it only uses common ancestry established through DNA/molecular evidence as a basis for phylogeny. So we're only paying attention to genetic and to evolutionary relationship, okay? And in cladistics, we make phylogenetic trees like this. They're called cladograms in that case. In a cladogram, the distance in between represents proportional distances of time. All right? Each group is called a clade. That means it has a common ancestor, and all of its descendants are included in a clade. All right? So, for example, here we have a common ancestor and all of its descendants. So we would have a clade. And we have three types of groups. We can find three types of groups in a cladogram. A monophyletic group, a group containing a common ancestor and all of its descendants. So, the circle in red, that would be an example of a monophyletic group. There are also paraphyletic groups, or a group containing a common ancestor but not all of its descendants, and a polyphyletic group, a group containing species with different ancestors. Here, and I'll show you an example. So, here we use the same cladogram. Group 1 is a monophyletic group. It contains a common ancestor here, and 100% are all of its descendants. Monophyletic group. A paraphyletic group contains a common ancestor, right here, but not all of its descendants, not 100% of them. See, that group isn't containing G, which is a descendant. If it contained G, it would be a monophyletic group, but it doesn't. And a polyphyletic group, which contains a common ancestor and more than just its descendants. It also includes C, which is not a descendant of this common ancestor. Why is this important? Because it's important because we're trying to bridge the Linnaean system of taxonomy and the cladistic system. The Linnaean system for a taxon, like a genus, is only equivalent to a clade if that group is monophyletic. So, when we started looking at molecular evidence and those things, those groups like Animalia, like Mammalia, mammals are not a monophyletic group, meaning they all don't share one common ancestor, for example. So, the problem with the Linnaean system of using genus and family and things is they're not monophyletic groups. Not all of them, at least. They all don't include one common ancestor in all of their descendants. Lots and lots and lots of them are polyphyletic groups, and lots and lots and lots of them are paraphyletic groups as well. So, many scientists now are saying, why don't we just go to the cladistic view? And I, for one, agree with that. So, how are we going to construct a phylogenetic tree? First, we construct a phylogenetic tree based on two things. One is shared characteristics. A shared characteristic is a trait that a given clade shares with its common ancestor. So, for example, if this was a clade here, I'm circling in red, a shared characteristic would be a vertebral column. A derived characteristic is a trait that is present in a clade or species but not in its common ancestor. So if we're looking at this clade here, amniotic eggs would be a derived characteristic for a turtle and a leopard when compared to the clade including the salamander. So, when we make cladograms, we always pay attention to derived characteristics and use derived characteristics to partition the branch points. So each branch point in a cladogram or a phylogenetic tree will occur after a derived characteristic has arisen. And those organisms finding themselves present or included in the cladogram after that derived characteristic has that derived characteristic evolutionarily speaking. So you might have noticed from this slide there's multiple ways using, for example, this diagram that we can construct a cladogram or a phylogenetic tree. We want to do so, and scientists do so from molecular and structural evidence in order to maximize parsimony. And what that means, they create these phylogenetic trees to maximize the fewest number of evolutionary events possible. So they are like the fewest evolutionary events needed to go from an ancestor to descendant represents the most likely sequence of events. Occam's razor, the simplest solution is usually the most correct one. Or if there's if there's multiple possibilities, probability favors the most simplistic of them. So, for example, if we're giving this data, there's 3 different ways that we can account for these evolutionary changes at different chromosome or at different gene sites. We can create 3 different phylogenetic trees. The one that is favored or considered the most right is this one, because this one postulates only 6 independent evolutionary mutations or events have to happen to go from an ancestral species to all of the descendants. These other 2 require 7. So to maximize parsimony, we're going to pick 6, the 6th one. Phylogenetic trees can also be used as hypotheses to test predicted morphological or genetic similarities between existing species based on shared ancestry. So, we can now create a cladogram or a phylogenetic tree and use the predictive power of it to go back and look at actual species that exist now that our cladogram predicts there may be structural similarities between, and then go back and look for those evolutionary or structural similarities. So there's two practice problems or two things I want to walk you through that we'll do more in class on Wednesday with, but they help us make and interpret cladograms or phylogenetic trees. So, number one, we're looking at this cladogram. In the following cladogram, which node occurred earliest in time? So all of our letters are going to be the most recent, and we're moving backwards. So, point 1 would be the earliest in time. 1 is the earliest. Which, in the diagram for question 1, which node represents the most recent common ancestor for terminal taxa B and C? The most recent common ancestor is 2. Most recent common ancestor is 2. Right here, there was a common ancestor, and it diverged and led to the creation of taxa 2 and of taxa C and D. Which terminal taxon is B more closely related to? A or C? This is what you need to think about. So, something happened here at 1, and something happened here at 3. And so B will be the most closely related to A, like that. Of the cladograms shown below, which one shows a different evolutionary history from the others? So which one can't we just rearrange or flip around the lines to look different? All right? So what you might want to do is pick and try to draw all of them out and see which one shows a different evolutionary history. So we'll start at the bottom. Number 1, we have A branching off first, number 2A, 3A, A. The rest go to B. B is next. B is next. B is next. Then we have C coming off from B. C coming off from B. C coming off from B. C coming off from D. No. So 2 would be the one that shows a different evolutionary history. Different evolutionary history is shown in the one that is circled. So, problem 2 says construct a phylogenetic tree from the below morphological data, whether they have vertebrate, bony skeleton, 4 limbs, amniotic egg, hair, or 2 post-orbital fenestrae, or holes. So what I'm going to do is I'm going to first draw a long line like this. I'm going to number these traits. 1, 2, 3, 4, 5, 6. Here is our common ancestor. Here's the most recent. They all have vertebrae. So vertebrae must have, if it is a shared trait, it must have developed first. All but one have a bony skeleton. So, oops, my bad. All but one had a bony skeleton. So, the one that didn't have a bony skeleton were sharks. So we're going to bring it over here, and I'm going to say this is sharks, and 2 must have developed right here. Now, go to 3, 4 limbs. Well, neither sharks nor ray fin fishes have 4 limbs. So, ray-finned fishes must have diverged before the development of four limbs. So I'll just put ray there. All right. Then there is amphibians. Amphibians, next, don't have an amniotic egg. The rest do. So, amniotic eggs, 4, must have diverged, or amphibians must have diverged before amniotic eggs. All right, so we have done 1, 2, I counted 3. All right. Next, we see hair. Primates and rodents have, but none of the others have. And we have 2 postorbital fenestrae, which crocodiles and dinosaurs have and none of the others. So what I'm going to do is I am going to have an offshoot that looks like this. And on this offshoot, we're going to have primates and rodents. And we'll have another offshoot that have crocs and dinos. And what we're going to have here is we'll have 5 be right here and 6 be right here. So that's how we would assemble a simple cladogram, something kind of like that. There's many ways we can make it look, but that might be one example of a cladogram. We're going to have more practice and go through some more cladograms and some phylogenetic trees next time.